a YouTube star, if or whether you consented to that or not.
Yep.
Yep, good.
Wait a few moments now. When people come in, so
We've got
a few, they're still maybe coming in. So welcome everybody to Imaging ONEWORLD. This is the second lecture in the year 2022. I'm Stephanie Reichelt. I'm at the University of Cambridge. And you know Alex Sosik, also from University of Cambridge, Nick Barry from the MRC Laboratory of Molecular Biology in Cambridge, and then our colleague Kirti Prakash, who can't join us today, but usually is extremely super active. He is now in London. And we also have Alessandro Esposito as the, also from Cambridge. Sorry, this was started in Cambridge as an online educational tool for anyone interested in imaging. And I think it's been really popular and many people are regulars. We have support from the Royal Microscopic Society, who helps us with the administration, and also we use their website, obviously, and they administer the website and send out all the information. So thank you, Jess and Georgina. Yeah, and so anyone I wanted to say who wants to suggest speakers who find you know, a particularly useful, really exciting, entertaining, educational lecture on imaging would like to provide, or know someone who could, please let us know and just email us to the RMS address, which also sends you the invitation to the lectures. But today my pleasure is to introduce Jörg Enderlein from the University in Göttingen, Georg August University Göttingen in Germany. Jörg has always been one of those people who gave lectures in the imaging community and conferences, which were not just showing extremely interesting and very challenging research, high-end state-of-the-art developments, but also I think he's an extremely good educator. So when you go to his website, you see that he has actually under Science and Fun and Teaching some fantastic tools where there are very interesting visualization of the physical concepts underlying the imaging he's also talking about today. And I think that is an amazing skill, which yeah, is really to your credit. And I mean, extremely helpful. And I mean, I think it's unusual. So it's also, I mean, there are other people who do it, but you do. I mean, I just looked at some of them and I was like, wow, this is fantastic. And that's something I hadn't actually seen before from your website. But Jörg came from Odessa, where he studied physics, and then he went to Berlin. And he was a postdoc in California, Los Alamos National Laboratory for a relatively short time, but then returned to Germany to the University of Regensburg, and he had a Heisenberg Fellowship from the Deutsche Forschungsgemeinschaft at the Forschungszentrum in Jülich, and then continued as professor in Tübingen, professor for biophysical chemistry, and since 2008 he's professor for biophysics at the Georg August University in Göttingen, and where he's running an extremely successful and very creative research group. His major interests are single molecule fluorescence spectroscopy and imaging protein conformational dynamics and folding. And he's also been a very long-term and early developer of FCS and contributed to the field, specifically what he's talking about today, I think, about metal and graphene-induced energy transfer imaging for the purpose of super resolution imaging, I think. And I'm very much looking forward to hear more about this. So over to you.
Yeah, thank you, Stephanie, for this very nice introduction. And I hope I can live up to the expectations. So it's my big pleasure today to be here and to talk about our recent work on metal and graphene-induced energy transfer. And before coming to this topic, let's simply start from the basics. So as you all know, when you have a wide field microscope, there's a resolution limit, which is simply telling you, okay, you cannot resolve something which is beyond or below some critical value. And to understand this resolution limit, I always try to present this kind of movie here, where you see the coherent emission of 2 point emitters. So there are 2 point emitters here at the very center, and they are emitting light. And what you see is electric field distribution around these 2 point emitters. And what is happening when they are coherently emitting, they are forming this kind of interference pattern. So these dark lanes are the lanes of destructive interference, where basically no emission takes place. And when you now imagine that an objective of your microscope is looking at these 2 emitters, what it sees is simply this kind of wavefront here, these electric field wavefronts hitting the front lens of your objective. And the animation is showing how this pattern is changing when the distance between these 2 emitters becomes smaller and smaller and smaller. So what is then happening is that these destructive interference lanes are moving to higher and higher emission angles. At some point, when the distance is too small, then the very first destructive interference is hitting the edge of your microscope objective. And then your objective only sees a continuous wavefront. It cannot distinguish anymore whether the light comes from 2 or multiple emitters or a single emitter. And by doing a very simple geometric reasoning, simply by asking, okay, what should be the smallest distance that the first lane of destructive interference is exactly at the half angle of light collection of your objective, you can then calculate, okay, this is exactly the resolution limit. It's classical resolution limit, which was found by Aber already in the 19th century. It tells you, okay, if the distance between 2 emitters is smaller than the wavelength of light, divided by 2 times the refractive index here in your medium and sinus theta, where theta is this half angle of light collection of your objective, then your objective loses the ability to distinguish between these 2 emitters. There is a second resolution limit, which is not so well known. It is along the optical axis. Of course, when you use, for example, a confocal microscope, where you scan with a focused laser beam your sample in 3 dimensions, and then you record at each scan position the intensity. There is a resolution limit for the third axis. And by doing a very similar reasoning using 2 emitters and looking, okay, how does the interference pattern look like around these 2 emitters along when they are placed along the optical axis, you come to a similar resolution limit for the axial resolution, which tells you again, okay, when the distance between your 2 emitters along the optical axis is smaller than the wavelength of light divided by refractive index, and now one minus cosine this half angle of light collection of your objective, then again, you cannot resolve them anymore. And to get some numbers here, this next image is showing you, the next figure here, how these resolution limits look for real microscopes. So as a function here of the numerical aperture, this is this refractive index times sinus this half angle of light collection I was talking about before. And typically when you have a water, let's say water immersion objective, so which is adjusted for looking into water or something like close to water cells and tissues, then we are talking about typical apertures between 0.5 and 1.2. And then you can see here also dependent on the wavelength, of course, how your lateral resolution or your axial resolution is behaving. So in the best cases here, what you see for a really high NA objective, you get down to something like 200 nanometer, but then it's the end of the story. Or here for the axial resolution, we are then in the order of something like 600 nanometers. And as you can see, of course, there's a huge discrepancy between the axial resolution and lateral resolution. So when you look at typical images, 3D images taken with a confocal microscope, you will always see that the image, the final image, looks somehow elongated along the optical axis, because the resolution along the optical axis is by a factor of 3 to 5 worse than the lateral resolution. Interestingly, I mean, even when you now go to the realm of super resolution microscopy, and I guess you have heard about all these new methods like stat microscopy or single molecule localization microscopy or other methods, still this discrepancy between the axial and the lateral resolution is still taken over to the super resolution realm. So also in all these blue methods here, we have a difference between axial and lateral resolution on the order of a factor of 3 to 5. Also, of course, our lateral resolution is much better than with a normal wide field microscope. Yeah? There are exceptions to the rule, and these are interferometric methods like interferometric, PALM, or so-called isostat, They are using two objectives, looking from two sides on the sample. They really use the interference of light in detection and emission. And they can come down to something like 1 to 5 nanometers, even along the optical axis. There's the recently developed method 3D Minflux from Stefan Hell's lab here in Göttingen. But I mean, they are marvelous techniques, but they are super challenging from a technological point of view. So not many people have them available, and they are really difficult to operate. So we were thinking maybe to come to some simpler ideas, but before coming to that, I want simply to mention that, of course, one big challenge also to these methods here is something which we call spherical aberration. This is shown here. So spherical aberration means that your optics, your optical system, let's say when you focus light into some single spot, that you have some aberrations which are focusing light coming from higher angles to a different focus spot than light from smaller angles, as shown here in this very simple geometry. This is especially difficult when you have a refractive index mismatch between your objective or your microscope and your sample, which is often the case. For example, when you use here and on the right hand side a water immersion objective, which looks into a cytosol with a very slightly higher refractive index in water than what you can see here now. When you look deeper and deeper into the sample, here it's 4 micrometers, here it's 8 micrometers, here 12 and 16. You see that your point spread function of your confocal microscope, which is shown or visualized here in these images, is getting more and more elongated. And also, what is more important here, that then the position where you see your emitter, here, for example, in this case where you have the emitter at 16 micrometer deep into your sample, that the apparent center position of the emitter is somewhere at 17.5 micrometer. That means you are mismatching, you are mismeasuring the axial position of emitters due to this refractive index mismatch and due to the spherical aberration. So we were thinking maybe to come up with a method which is free of these problems and which is also much more accurate. And one way is to use so-called near-field optics or near-field fluorescence. So one very classical method of near-field fluorescence, and I guess you have all heard about it, is total internal reflection fluorescence microscopy, where you excite your sample from the glass side with an incoming light beam at a very high incidence angle. The incidence angle is so big that you have total internal reflection of incoming light on the glass water interface, so you have basically no light penetrating the water side. However, there is still an evanescent field above the glass interface in the water side. And what is shown here is the excitation intensity, or basically the light intensity of this evanescent field as a function of incidence angle. So for an incidence angle here, 64 degree, which is close to the critical angle of total internal reflection, you have still an exponential decay, but a very long penetration depth into your water, into your sample. When you increase here your angle, incidence angle of your incoming light, then this kind of exponential decay becomes steeper and steeper. And the idea now, when we are going back to our original problem, how can we measure the very high resolution on the optical axis? One clever idea, which is called variable angle total internal reflection fluorescence microscopy, is now that, let's say you have a sample, you have some fluorescent labeled molecules or structures in your sample close to your surface, and you want to measure the distance of your structures, of your molecules, with a very high accuracy. What you can do is you can take images, different images in total internal fluorescence reflection modus, but using different incidence angles, so that you modulate here the penetration depth. And when you take different snapshots of the same sample with different incidence angles and different penetration depths, then I hope you can somehow imagine that by having these different images, you can back calculate then the exact distance of your object from the surface by simply looking how much or how quickly it is fading away when you increase the angle. So when something is very close to the surface or directly on the surface, you wouldn't see any change in intensity when you change the angle. When something is far away, let's say here 300 nanometer, and of course when you increase the angle, then the intensity goes down very quickly. So this is the idea of variable angle total internal reflection fluorescence microscopy. Indeed, with such a method, I can localize fluorescent molecules with something like 10 nanometer accuracy along the optical axis. However, of course, the downside is you need a turf microscope, and you need a very precise variable angle adjustment. And also, I mean, it is intensity based, so it's not so easy, I mean, as it looks here, but in principle it works. But There's a second way. You can also use Nearfields to come to the same result, but in the emission on the emission side. So this is excitation. Now let's look on the emission side. So what is emission? So when you have a fluorescent molecule, you can very nicely model this molecule as an ideal electric dipole emitter. And this is classical electrodynamics, but it is very well describing the reality even on a single molecule level. So a single fluorescent molecule, as I say, it can be modeled as an oscillating electric dipole emitter. So you have a positive and negative charge, and they are oscillating around the given axis. This is the dipole axis. Of course, there's an oscillation amplitude, and there's an oscillation frequency, which is, of course, the frequency of light. And when you put something like that into Maxwell's equations, classical Maxwell's equations, then what comes out is the classical Hertz dipole emitter. So what you see here now is simply model calculation of the electric field around such an electric dipole emitter. Of course, this animation is slowed down billions of times. Yeah, this is, of course, not real time. In real time, the oscillation frequency and also, of course, the frequency of this light emission would be something like 150 terahertz in the visible spectral region. But in principle, this is exactly how the electric field looks like around a single fluorescent molecule when it's emitting. So this is classical physics, nothing new, something simply let me explain you one specialty in this animation. If you look here along the axis of your dipole, so this is the dipole orientation. If you look along the dipole axis, then you see that in the far field here, there is basically zero electric field. So there is no far field emission of your molecule along its direction of its dipole emission dipole. The maximum emission is in the equatorial plane here. And this is also what you then really can measure when you have a single molecule in space. Let's say you can fix it and you would simply measure what is the intensity I see when I look from different directions. You would see zero intensity here along this axis, and you would see maximum fluorescence intensity along this axis. This is classical physics, very well established, so nothing new, but the fun starts when you now take your molecule and you put it very close to an interface. This is shown here schematically. So here's an interface. Let's say it's interface glass, the cover slide here, and your water is here, and here's your single fluorescent molecule with some given orientation. What is now happening is that your molecule is emitting energy or photons in different directions, and they can partially reflect from the interface because there's no refractive index mismatch. So there's partial reflection. For example, air-glass interface, you would have something like 4% reflection intensity for 90-degree incidence angle. And this partially reflected light, this is now the fun part, can then back react on the emitter itself. You know, although it's a single photon emitter, this photon somehow is virtually split, can split and can back react on your emitter that can modulate its own emission. Now, this is then really quantum optics. So it's single photon self-interference or single emitter self-interference. The mathematics, I will not go into details here. This is also very well worked out, was already in the last century by Chance, CRUK and Sylvie and other people, Lukosch and Kuhn and Drexhage. And one very funny modulation of this emission is that we completely change the energy which is emitted into a given direction into space. So as I said, when you look at the Hertz dipole emitter, there was zero emission along the optical axis, there was maximum emission in the equatorial plane. In between is a very simple sine squared theta law, where theta is the angle between the dipole axis and the axis of observation. However, now when you put your emitter on this glass, let's say border interface, everything is changing. So what this animation is now showing you here is how the angular distribution of emission is changed due to this glass interface. So this is an animation for a molecule is sitting in air on a glass interface. And what you see in this animation is simply that I changed the inclination angle of my dipole emitter with respect to the surface. The same here on the right side, only here my emitter is all the time with its dipole axis in the glass air interface, but it's rotating around the optical axis. So what these figures are showing is how much energy is emitted from your emitter into a given direction. You see, I mean, this is now a very complicated figure, and it's indeed dependent on the relative orientation of your dipole with respect to this interface. Can we somehow measure that? And this is also the next fun fact. Yes, I mean, the single molecule fluorescence emission today and detection possibilities, we can measure that. So what we do is we are imaging individual molecules with a normal wide field microscope, which is sensitive enough to see single molecule fluorescence, but we are defocusing our microscope on purpose. So instead of trying to have a very sharp focus, we are slightly defocusing it so that the light which is emitted into different directions is somehow distributed over a larger area on the detector. And then what to expect from these numerical calculations is this kind of so-called defocused images or defocused patterns of single molecule emitters when they change orientation sitting on an interface. And one of the most beautiful realizations of this experiment were done by Hiroshi Ushi, who is a university, he was a professor now in Leuven, also in Japan. He has a split professorship. And what you see here is now an experiment what he did many years ago, where he embedded single parylene molecules with a very sparse concentration into a super thin polymer film, so that the molecules, they cannot move away, they cannot diffuse away anymore. They are trapped in nano cavities in the polymer, however, they can still rotate. So what you see here, each of these patterns is a single fluorescent molecules, and the motion what you observe here in this animation is the reorientation of the molecules. Yeah, for example, some of them are quite fixed in orientation, like this one here in the top left corner, but some they rotate very quickly, like this one here, and then you see indeed all the different patterns for vertically oriented molecule or in plane oriented molecule and so on. And we use this method and in many different publications for indeed reading out the rotational diffusion dynamics of single molecules, or for example, looking at bioproteins which are reorienting in space and then using fluorescence to watch their reorientation. So I mean, this is not the main point here in my talk today. So there's a second very interesting thing. So again, let's look at the single emitter on the surface, and let's say we look at a random oriented emitter, so we simply average over all orientations of our emitter. So again, what you see here is a cross section across this angular distribution of emission. And as you have seen before in the animation here, indeed, there's this kind of emission peak. By the way, this emission peak is exactly at the angle of total internal reflection between the glass and the water side. And then this is basically, this is then the angular distribution of emission down to the glass side. There's also, of course, emission towards the water side. This is this green guy here. But there's now something really interesting. You see that there's some considerable emission above the critical angle of total internal reflection. And the fact is, when you now remove your molecule away from the sample surface, then this so-called supercritical angle fluorescence, the red part, will go down and down in intensity. So it will get less and less. The blue part will be completely unchanged. So that means when you move away your molecule from the surface, then this blue part, the blue emission part here, will remain unchanged, but the red part will gradually go away. And by measuring the intensity of fluorescence, which is emitted above this critical angle here in this red region, and you compare it with the emission intensity of the fluorescence emission in the subcritical range here, the blue part, you can then indeed back calculate the distance of your emitter from the surface. This is called supercritical angle fluorescence emission, or microscopy, submicroscopy. And what you see here in the figure is simply the ratio of the supercritical fluorescence intensity versus the total fluorescence intensity. As you can see, indeed, when I move my molecule here away in the order of a few hundred nanometers, and this supercritical angle is completely decaying. And this is again a near-field effect, because that the molecule can at all emit in the supercritical angle regime is indeed the near-field coupling of its near-field electric field into the glass half sphere, which is a quite complicated physics. So I will simply say it here that it's a near-field effect, a near-field coupling of the near-field of the emitter into the glass half space. So this is now, by the way, quite successfully even commercialized by Sandrine Ledec-Four in France. So she's publishing wonderful work using this method, and indeed she can achieve here localization accuracy of a few nanometers using this method. We were then think in a similar direction, but instead using intensities, we are using the lifetime, because the second important parameter which is changed when you have a molecule and you approach an interface, which is a refractive index mismatch, is also the fluorescence lifetime. And to increase this lifetime effect, we not only use something like a glass-water interface, we are covering our glass substrate with a very thin film of metal. And then you see a huge modulation of the fluorescence lifetime as a function of the distance of your emitter towards the gold or the metal surface. So why is there now such a super strong dependence on distance and lifetime distance dependence? Because what is now happening is the following. Your molecule, as I said, you have this electric field around your emitting molecule, and there's a very strong near field component. And when your molecule comes very close to the metal, you have to remember in the metal, you have basically a free electron gas. Then these free electrons in the metal, they can sense the near field, electric near field of your emitting dye. And then these electrons then start the surface plasmon, and they can grab the energy from your molecule and then transfer it away into so-called surface plasmons. And this strong coupling leads then that the molecule can return to its ground state much faster than before. And this is then seen here in this very strong modulation of its excited state lifetime. The beauty now is that we are completely independent on intensity measurements. So what we can do is simply we take our emitter, the image with a fluorescence lifetime imaging microscope. I show you in the next slide. And then we know the lifetime that we can use these curves here to back calculate this distance from the surface. Of course, what is also seen here, this effect, in the same way also as before, the soft microscopy is orientation dependent. So theoretically, you should know exactly how your molecule is oriented with respect to the surface. So for vertical dipole, the green curve, you see a different dependence on lifetime versus distance. And for the horizontally oriented molecule, fortunately enough, in most of our applications, when we do bioimaging, then we see that our molecules, because they are coupled to structures of interest with a very flexible linker, that you can use the random orientation because the molecule is reorienting all the time in space, and that's why the so-called random curve for the isotropic emitter is an excellent approximation of the true situation. So how does it now work in microscopy terms? So this is now shown here, and we call this method metal-induced energy transfer or MIT imaging, because the physics basic what I'm talking about here is this energy transfer from the molecule to the metal. So it's a metal-induced energy transfer, and the name is very similar to FRET, first the resonance energy transfer, where you have a very similar physics, but the energy there is not transferred to a metal, but to another molecule, but the physics is very similar. So what we now do is the following. Let's say you have a cell and you want to profile the basal membrane of the cell with extreme high axial resolution. So what we do is we are labeling the cell membrane with a fluorescent dye. This is nothing special. You can buy these dyes from different companies. But then the big modification is that we are now putting our cell on a glass substrate which is covered with a very thin metal film, typically 20 nanometer gold film. And the gold film is so thin that it is basically transparent. So when you look with your bare eye on such a cover slide with 20 nanometer gold, you will nearly not see anything because it's transparent. It's so thin, it's transparent. However, the presence of this very thin gold film is enough to induce this quenching here, this fluorescence lifetime modulation, what is shown here. And then, of course, you need a microscope which can measure not only the intensity, but also the fluorescence lifetime. And what we use is a confocal scanning microscope, where we have here an excitation laser which is pulsed. This is something like 20 megahertz pulse repetition rate and a few picoseconds pulse length. And then we have here an optical tunable filter for picking the right wavelength. So this is a white light laser. Here's a wavelength filter simply. Then we focus the light into a very tiny spot here onto our sample. And then we scan. We have a scan unit. We can then scan our cell here. And then at each scan position, then we detect single photons here with a very sensitive photodetector, which is also super fast in its response time, so that we can then time the photon arrival times with respect to the excitation pulses. And this gives us a fluorescence lifetime information. So this is called time-correlated single photon counting. I will not go too much into detail here, but it's a standard method when you do a fluorescence lifetime imaging microscopy. So this is all what you need. You need a standard film microscope, and you need this one special cover slide with this gold fin. That's the only modification with respect to a normal film microscopy approach. So now we can measure intensities in lifetimes, and then we can use our perfect knowledge of the physics behind to convert the fluorescence lifetime into a distance. And the first publication is now already 8 years ago. This is what you see here. That was then a living cell, an MECK2 cell. It's a cancer cell. And we labeled with a fluorescent dye the plasma membrane. What you see here is now in basically quasi-life cell imaging, the dynamics of the basal membrane of the cell when it moves around the surface. And you can see in the false colors is shown the distance between the membrane and our substrate. And you can easily see, yeah, I mean, we are now talking about an X resolution here of nanometers. And I will quantify it in the next slide. Importantly, of course, the lateral resolution, the XY resolution, is still diffraction limited because it's a normal standard conformable laser scanning microscope. However, now using this lifetime trick and the metal film, we get now extremely high X resolution. We then use this method for measuring the distances between between the membrane and the substrate for different cell types. This is shown here, and the nice thing is that there are alternative methods using interference in a more complicated manner, and they came to the same conclusions. And this is really nice that we could confirm what people have seen with more complicated methods before. Now, of course, the question was, okay, how accurate is our method? How good are we in accuracy? So what we did is a test measurement where we simply put on our gold glass substrate spacer, a silicon dioxide spacer of a very well-defined thickness. So we have in a clean room, you can prepare such samples very precisely. And then on top of the spacer, we have a super thin film of fluorescent dye. And then again, we basically repeat this experiment, this mite imaging experiment many times. And then from the variance of the result, we can then estimate what the accuracy is. And what you see here on this graph is the X resolution of our method as a function of the fluorescence intensities in 1,000 photon counts. So it means when you collect something between 5 to 10,000 photons, then the accuracy for the X resolution is between 2 to 4 nanometers. And this is, I think, some really remarkable number. Then typically, this is the range where when you have a normally well-prepared sample, this is the typical range of fluorescence intensity what you have in a good labeled sample. So we then apply this method for different biological applications. This is here a collaboration with the group of Andreas Janshoff in the chemistry department. They are, for many years, they are studying this epithelial to mesenchymal transition of these epithelial cells. So they can trigger this transition. And then what we did, we labeled the actin cytoskeleton with a fluorescent dye and then used mite imaging to watch the axial distance, the average axial distance of the actin cytoskeleton from the sample surface. And as you can see here, comparison between our translating cell and the non-trivial cell, you see indeed that the actin cytoskeleton during the transition is somehow uplifted by some 20 nanometers before coming back again then after the transition. And of course, something like such subtle changes would be super hard to see by other methods. And the good thing, of course, I have to emphasize it again and again, that mite imaging is perfectly live cell compatible. So this is also a big contrast to many other methods. Of course, you don't want not only to see a single structure, you want to see several structures simultaneously and to do some kind of co-localization. So then we made the second step, the next step, we introduced dual color metal-induced energy transfer imaging. So by simply labeling different structures with different dyes, and again, then doing the same trick, measuring the lifetimes and then determining distances from surfaces. And what you see here is now proteins in the focal adhesion complex. And we labeled proteins here in the focal adhesion complex, actin bundles and vinculin filaments. And you see very nicely these stress fibers from the focal adhesion complex for different cells or different cell positions. And again, we have here then the distance already converted to distance values. And you see here that the actin is more reddish and the vinculin is more bluish. And indeed, when you then simply follow individual stress fibers, then you see clearly, yeah, the actin bundles are always above the vinculin, the corresponding vinculin protein fibers. And the distance between them is an order of 20 nanometers, again, very small distance. And also pay attention that we can measure very subtle height changes over a super long range. So this is 9 micrometers. So we are following here one stress fiber over 9 micrometers, and we see then a height change from 30 to 50 nanometers. Again, this is something which would be very difficult to do with other methods. One of my favorite examples or applications is the nuclear envelope mapping. So as you may know, the nucleus in our cells, they have a double membrane. It's an inner nuclear membrane and outer nuclear membrane. It's a double membrane system similar to mitochondria. And the distance between the inner and the outer nuclear membrane is again super small, beyond any normal diffraction limit of optical resolution. So what we did again, we used now here LAP2 beta proteins inside the nucleus at the inner nuclear membrane to label them with a green dye, and we used one protein from the nuclear pore complex to label with a red dye. And again, we did this dual color measurement, and I will simply show you the result. This is shown here. Let's first look at the left side. For 6 different cells, this is the distance that we then measure between the inner and the outer nuclear envelope. And we find consistently a distance between 35, 40 nanometers. More importantly for me, at least as a physicist, was that we can then switch the order of the dyes. So in the beginning, as I said, we had the blue dye inside and red dye outside. Then we switched the order, we put a blue dye in the outside, and we took the red dye for labeling the inside. And we find back the same numbers. This is super important because it tells you indeed that this MIND imaging is a bias-free absolute measurement method. So there is no systematic bias, whatever. And we don't need any reference measurements. It's all basically fundamental physics which goes in. That's all what you need. Of course, and the next question we were asking is, okay, how sensitive are we? Of course, we have this metal film here in a gold film, 20 nanometer. As I said, it's basically transparent, but still you lose light. So I mean, when you do the exact calculations, then you can estimate that something like 30% of the emission light from your molecules is lost in the gold film, simply absorbed in the metal. So the question was, okay, is it still possible to see single molecules with our method? So then Narayan, at the time, my PhD student, he did the experiments. He again prepared some very well-defined test samples with a well-defined spacer here of silicon dioxide. And then on top of them, he positioned with very low concentration single molecules. And the result is shown here in the next slide. What you see here are 4 different measurements with 4 different distances between the molecules and the metal. So here's 20 nanometer, 30, 40, and 50. And the color here is giving the lifetime, fluorescence lifetime. So bluish means shorter lifetime here in this range. Reddish means you are here in the longer lifetime range. And what you can see, of course, the first message is here. The signal to noise ratio is still excellent. We can see with very high signal to noise contrast, we see single molecules. The second message is, and indeed, when you move only by 10 nanometers your molecules away from your gold surface, then it's a huge change in lifetime. So you can clearly see the difference. And when you do the statistics, again, we can estimate how good is the single molecule localization along the optical axis. And we find here for roughly 8 to 10,000 photons till photo bleaching, which is of course then the limit of a single molecule experiment, we find consistently a next resolution of 2.5 nanometers. So that was a very good message. The next question was, how fast can we go? So everything I have shown you before was done with a very slow scan system, which is using a piezo table for scanning the sample XY, which is slow. So for example, this live cell image from the beginning, it took something like 3 minutes for taking 1 frame. So it was really a slow method. Of course, we wanted to go much faster. And then fortunately, we came across a new development by Picocron. They developed a new XY scanner for very fast confocal scanning imaging. This is called the Flimmy. This is shown schematically here. And with this kind of imaging system, now we can go up to something like 10 or 20 frames per second. And here again, you see now a typical example of actin filaments, labeled, this is the intensity, this is the lifetime. So we can now take super rapidly lifetime images. And one application, of course, now in the context of MITE is shown here. So this was a collaboration with Sarah Köster from our department. She is interested in this blood platelet spreading or blood platelet dynamics. So blood platelets are super important for blood coagulation or when you have a wound or whatever. And what is happening when you have these blood platelets, which are without the nucleus, when they are approaching a surface, then they start to spread extremely rapidly. What you see here and on the left side in the intensity image. So this is basically what you see from the start is when a platelet is settling on the surface, it starts to interact, it's spreading extremely quickly, and also the whole actin cytoskeleton of this platelet is then reorganized. And we can then watch this reorganization along the optical axis again with our MITE imaging. So what you see here now is already the lifetime image converted into a height image. There are some black spots here in the middle because here the distance is too far for our dynamic range. As you have seen, there's basically a dynamic range of something like 150 to 200 nanometers. We cannot go much beyond. And here in this range, here in this experiment, we are limited to something to 50 nanometers. But then again, you see that now with this very fast fluorescent lifetime imaging, we can now follow the spreading of platelets with a very high temporal resolution. Of course, now the question is, can we combine our method with the classical method of super resolution for getting also a high lateral resolution? Because everything I'm showing here is a fantastic axial resolution, but still the XY resolution is the normal confocal laser scanning microscope. So the question is, can we now somehow extend our method to also do super resolution along the XY direction? And the method of choice for us, of course, is so-called single molecule localization microscopy. I hope you are familiar, you heard about it before. The idea is quite simple. So when you look again at the image of a single individual molecule on your wide field microscope, then of course this molecule appears much bigger than its physical size. Now, the normal molecule has a size of something like half a nanometer in diameter. But here in the image, when you look in the normal wide field microscope, then the image of a single emitter, single molecule appears something like 200 nanometer wide. This is exactly the resolution limit. So this is the manifestation of the resolution limit, this broadening of the image of a super tiny emitter. However, if there is only a single molecule somewhere in one patch of your image, and you know there's nothing else around, then of course you can still fit the center position of this huge wide intensity distribution. You can fit the center position with an extremely high accuracy, much higher accuracy than the width of this distribution. And when you somehow can come up with an idea or method that you have only a few molecules switched on at each time, and you look at your sample and you can localize these molecules in this manner, then you can sequentially build up an extremely high resolution image of your whole sample. This is the idea of single molecule localization microscopy, and of course the most prominent representatives are PAL microscopy, as a photoactivation localization microscopy, and stochastic optical reconstruction microscopy, or STORM. So we then combine this with our lifetime capability. So what we did here now is, it looks like a standard single molecule localization microscopy experiment, but it is not, because what we do now here is we use our very rapid scanning system for taking with very rapid succession scan images. And then we treat these scan images as if they would come from a normal wide field microscope in a normal STORM or PALM experiment. So the difference now here, this is not a wide field image, what you see here in this movie, these are scan images. But still, we can now also in these scan images, we can localize individual molecules. We can fit their center position with very high accuracy. This is shown in here, the comparison between the super resolved image reconstructed from single molecule localizations versus the normal confocal image. The same here for here. We use here another map, which is called PAINT. I will not go too much into detail. It's very similar to STORM PALM, but it's even more clever and gives you even higher resolution. So this is here chromatin in the nucleus. And again, here you see the comparison between the super resolved image and the normal confocal laser scanning microscope for resolution. But now, because we have here the scan images, we can also now add a lifetime dimension. This is very nice. For example, here you see the lifetime distributions of all these molecules in this sample, and here you see the lifetime distribution of all the molecules which we identified here in this sample. And you see, of course, a clear difference. So you can basically now do something like a quasi-multicolor imaging using this method, the same emission color. This is quite important because, I mean, let me explain it here, because the chromatic aberration is a big issue in super resolution microscopy. So what is chromatic aberration? And when you take a normal single piece lens, what you will observe is that when you shine white light on it, then different colors are focused at different positions. Now, of course, nowadays we use compound lenses in our modern microscopes. They use different glass sorts to compensate for this chromatic aberration. But this compensation is never 100% perfect. And especially when we are talking about some resolution in the nanometer range, which is now easily achieved in single molecule localization microscopy, then chromatic aberration again becomes a big issue. The good thing is when you use lifetime for distinguishing different molecules which are emitting at the same emission wavelength, then you can completely avoid this chromatic aberration problems. And this, I think, is one very nice application of this scanning lifetime single molecule localization microscopy, or as we call it, the FL-SMLM. However, now let's come back to our mite story. We now can combine everything into one system where we use the mite, the metal-induced energy transfer, for getting the axial position of a molecule, and we use the laterally then normal single molecule localization microscopy to get a high lateral resolution. And this is now very first data which are just submitted. Here you see now microtubuli here in the slide, fixed microtubuli, and we have labeled them in fluorescent dye. And these are cross sections, XY cross sections at this position, at that position. Of course, the data are very noisy. We are talking about single molecule data, and when you take this cross section, there are not so many molecules in this given position. But I think you can still see that indeed we see the hollow structure of the microtubulus, and indeed also the size dimensions what we find here are perfectly corresponding to what we expect when we take into account the diameter of the microtubulus and an extra linker length and the size of the dyes and so on. Then we get exactly what we expect from the cross section of these images. So I think this is now very nice method to have the best from both worlds. We get super high resolution with mite, long optical axis, and we get super high resolution laterally with standard single molecule localization microscopy. But still, this was not the end of the story, because we wanted to even go further. And this story, what I want to tell you next, was initiated in our lab by Falk Schneider, who was at the time visiting student from the lab of Christian Eggeling in Oxford. He is now a postdoc in California. And the idea was he wanted to, the question what he wanted to answer is, okay, is our method good enough that we can even resolve something like a single bilayer? As we have seen, I mean, I claimed that we can somehow localize single molecules with 2.5 nanometers. So a single bilayer, like a plasma membrane bilayer, has something like 5 nanometer thickness. So theoretically, you would say, yeah, this should be possible. However, the question or the difficulty is not the following. When you want to resolve this bilayer, you have to label the bottom and the top leaflet. And normally we do it here by simply labeling the head groups of our lipids. And then in one measurement, when you take now a fluorescence lifetime measurement of such a system, then you have two different fluorescence lifetimes, one from the dyes here at the bottom and one from the dyes here at the top. But for 5 nanometers, the lifetime difference here is only something like 100 picoseconds. And for a compound signal, so when you have a mixture, bi-exponential signal, where the two components are differing only by 100 picoseconds, this becomes really challenging now to disentangle. So using a metal like gold, it will not really work out. So we tried it and it was a failure. So we were thinking, can we improve our method to get to the super high resolution, even when you have simultaneous labeling in two different distances from the surface? And the solution in the end was to use graphene. So we found a paper from our colleagues in the Netherlands, Buchenboom and colleagues. They were showing very nicely that when you use graphene, you can induce the same lifetime modulation, as a distance lifetime modulation in single molecule emission, but on a much shorter time scale. So again, what you see here is the fluorescence lifetime as a function of distance. But now for the interaction of a single emitter with a single graphene layer, so graphene you may know is an hexagonal array of carbon atoms, and we use the simply graphene on a glass slide, and we have again here the spacer layer. And now you see that this interaction range is much shorter than before. For gold, we had something like here, x-axis, up to 150, 200 nanometers. Now for graphene, the interaction range is in the order of something like 25 to 30 nanometer. What is also shown as a technical detail, I did not tell you so far, that of course all these curves also depend on the fluorescence quantum yield of an emitter. That means how well an emitter is converting absorbed energy into light. So quantum yield of 0.1 tells you, okay, only every 10th absorbed photons is re-emitted as fluorescence. Quantum yield 1 would mean every absorbed photon is re-emitted as fluorescence. But this is a technical detail, so it's not so super important, simply for the sake of completeness, I mentioned it here. So then again, Arindam, who's now a postdoc in Markus Sauer lab in Würzburg, he did a very beautiful experiment. Again, he used these test samples where we have a graphene layer, we have a spacer, now much smaller spacer thickness, 10, 15, and 20 nanometers. On top of them are single molecules again. What you see here now are fluorescence lifetime measurements for single molecules. That's why they are so noisy. So each of these curves is a single fluorescent molecule on such a substrate. And you can see very easily that indeed by shifting the distance of my molecule by 5 nanometers away from the graphene, we see a huge change here in the lifetime. And this was very promising. We said, okay, now we can do the real experiment. So we went back to the bilayer. We prepared a supported bilayer labeled on both sides densely with fluorophore. It's not single molecule anymore. This now here's experiment. So it's a densely labeled bilayer with a high density of fluorophores in the bottom and the top leaflet. We put them on our graphene sample, and then we took the measurement. So we measured the fluorescence intensity lifetime for a really long time so that we could collect a few millions of photons. And this is the result. Let's first focus on the left side here. It's a little bit complicated. So as I said, we have now these two leaflets. Both leaflets, they carry fluorophores. So what you now expect on a graphene substrate is a bi-exponential decay. And there should be a short fluorescence decay component from the fluorophores close to the graphene. And there's a long fluorescence decay component for the fluorophores far from the graphene. This is exactly what we see. What you see here now are exactly these two lifetimes which are fitted to the measured data. And this is the short lifetime component, the long lifetime component. But of course, if you have only a finite number of photons, let's say 10,000 photons per measurement, then you have a quite substantial spread in your fitting. This is shown here by the width of these distributions. This is giving you some idea how accurate the measurement is. However, when we repeat these measurements with more and more photons, let's say here then in extremum, then at 10 million photons, of course, the spreading, this uncertainty in a lifetime goes down to shot noise limit. And when we then convert these curves into distances from the surface, then we find here these two distributions. There's also a gap here in between. This is not an artifact. This is indeed due to that we are now so sensitive that the presence of the bilayer is already affecting the electrodynamics. So it really matters whether my dye is above the bilayer, which has also some its own dielectric properties, or whether the emitter is below my bilayer. And this is then making this gap here. But of course, the jump here is only a few angstrom. So now let me summarize what we then could extract from these measurements. So in the end, what we wanted to measure is the thickness of bilayers. So we did different support bilayers here, BOPC, BLPC, and these are, let's simply focus here on the difference between the bottom and the top leaflet. And we find here for the longer acyl chains, we find 5.5 nanometer plus minus 2 angstrom. And for the shorter acyl chains, we find here 404 plus minus 0.6 angstrom. Of course, this is already at the edge what we can do. But I mean, what is shown here by having enough fluorescence intensity and using graphene as the quencher, we can indeed measure now distance or distance differences with something like angstrom accuracy. And we compare these numbers with classical measurements from small angle X-ray scattering. This is the competing method. The numbers here are smaller, which they should be, because the SAX is measuring really head group to head group distance, whereas we are measuring the fluorophore distance. So there's an extra linker, and of course the fluorophore also has some finite size. That's why you always see that we are somehow 1 nanometer thicker than the SAX numbers. What is also quite interesting that we see a relatively large distance of the bottom leaflet from the glass support. We did not expect that there is such a huge difference because the distance, because it's basically only a few water molecules. But also this was confirmed by SAX measurement. Indeed, the lipid bilayer, even the support lipid bilayer, is somehow floating on a cushion of water. And this cushion has a thickness in the order of 1.8 nanometer. That means really a dozen of water molecules thick. Last step application, and I'm over here, and I'm finished here. This is one of the very last, very nice work by Arinder, mainly by Arinder Ghosh and his colleague Sufi Raja in Duke University. So Sufi is a big expert in mitochondria. So he provided us with single intact mitochondria preparations. And what we wanted to study now with MITE is the organization of the mitochondrial membrane. Again, mitochondria, as you may know, has an outer membrane and an inner membrane. The inner membrane is very rugged. Yeah, it's has these these kind of very, very rough and non-planar structure. So what we did is we labeled again the outer and the inner membrane with different dyes, and we put them on our graphene substrate. And what we wanted to see whether we see any change in the mean distance, it's the mean distance, of course, of these red guys here and the blue guys when we activate mitochondria. So mitochondria, as you know, they are the energy factories of our cells, and they do basically ATP synthesis by using a protein gradient as a proton gradient. And we wanted to see whether we can see any reorganization of this membrane structure when we activate them, when we switch them on. So this is the measurement. This is the raw data. It looks not so super impressive here. This is the inner membrane. This is the outer membrane. The scale is slightly different here, lateral scale. That's why it looks like different, but it's the same measurement. And here in the resting state and in the activated state. What you see here in the bottom, of course, you don't see any change from getting from resting state to activated state. However, here you can somehow already sense that there's something happening. Indeed, when we do then the precise quantitation, that we find that upon activation, the mean distance between inner and outer membrane is indeed changing by something like 2 nanometers. And this is really consistently seen over many different mitochondria. It's not an artifact. So we checked over and over again. We made lots of control measurements. This 2 nanometer change is really there, and it's very pronounced, and we have the method to see it with this graphene-induced energy transfer. So with this, I'm at the end of my talk. I simply want to hint you that if you want to do something like that in the future in your lab, this is all publicly available. So there's a MITE GUI, which you can use. You can download from our website. You can use it. You have to input your data, then you get basically the MITE image out from that. There's a fantastic book about the whole method. That is basically the PhD thesis from Narayan. So he won the best PhD prize of our faculty, and as a reward was a free printing of his PhD thesis by Springer. So this book is existing, and there's everything you want to know about the method. And I have to thank all my co-workers here in Göttingen. It's a huge team. Most of them here in the top, they already left our lab. Alexei and Anna are still here. And these are the current co-workers, which are all working in this direction. And let me use the chance here, also last but not least, to advertise a new journal by the Biophysics Society, where I have the big honor to be the editor-in-chief, and which is focused on new methods, new technology, new ideas for biophysics. So maybe the next publication you may consider this kind of journal. So thank you for your attention. That's all I wanted to tell you.
Thank you so much for such a wonderful talk. So now we will probably just get on to the quiz. which I'll just run in a sec. And then if anybody else has questions, please feel free to type them into the chat, and then we'll get onto them just after the quiz. So, Georg, if you could unshare, please.
Yeah.
Thank you very much. Now I can switch to the quiz.
Mache ich das denn jetzt hier freigeben? Scheiße. Anschneiden, stoppen. Ja. Sorry.
Right. You should see the opening. I've got the first thing of the
Okay.
quiz. So that was what is graphene. That was just to get people signed up. So if you look in the chat, you'll see the link for the sign up for the Mentimeter quiz. I see we've only got 2 people in, although we've got a few. So if we get a few more people joining, you should see the link in the chat.
Yeah.
And then if you just, then you should just get into this. Oh yeah, here we go. There's a few more people. Once we've got a few people, then we can just get into the quiz itself.
So I see one question by Alice Bender. Shall I answer it?
Sure, you can actually, yeah.
Yeah.
While we're talking, if you could just say what the acronym is. I guess it's fluorescence lifetime.
Yeah, this is fluorescence lifetime correlation spectroscopy. I did not talk about it today. This is one method we developed many years ago, and also Martin Hof and Prague, and Alice was part of it. It's a very cool method where you can do fluorescence correlation spectroscopy measurements. I hope you are familiar somehow with that. So basically what you look is at intensity fluctuations coming from a very tiny excitation detection volume. And then you can use this kind of fluorescence correlation spectroscopy normally to measure diffusion of molecules in solution or in a membrane. And of course, what we then developed many years ago is a method to determine fluorescence correlation curves in a lifetime dependent measure. So let's say you have a mixture of a long and a short lifetime, and then you can, in one measurement, by using all the lifetime information, you can generate 2 correlation curves for a long lifetime and a short lifetime component. And of course, now the idea is very near-lying that when you have, for example, a supported bilayer, where you have labeling in the top and bottom leaflet, and where the lifetime now, due to the graphene interaction, is different in the top and the bottom, that you can now use fluorescence lifetime correlation spectroscopy to look at the fluorescence, at the diffusion of dyes in the top and bottom leaflets separately. And indeed, yes, we have done these experiments, and they look very, very nice. We have simply to publish them. Yeah, I can simply tell you it works. Yeah, we use graphene-induced energy transfer for measuring diffusion in a leaflet specific manner, and it works very nicely. The problem why we did not publish yet is that we still are fighting with the model, because what we see is that the diffusion behavior is much more complex than we expected. Yeah, we see also some very interesting coupling of diffusion between leaflets, which is not so simple to explain. This is the only reason why we still did not submit it. But the method works.
All right, super. I think we could get on with the quiz. So here we go. We've got a few participants. All right, let's start the countdown. Okay, question 1 just coming up. What determines the spatial resolution of a microscope? You've got 15 seconds to answer. The focal height of the tube lens, the magnification of imaging, the wavelength of light, the absence of spherical aberration, or the numerical aperture of the objective. And we've got a few answers, and in this case we had 3 correct, more than 1 correct answer. So let's have a look at the scores. Coming up. Stingalicious, Ashley, and Hula Hop Boys, and neck and neck. Okay. Okay, I think it's anybody's competition at this stage. Let's go to the next question. All right, and here we go. Next question is, what is the most important characteristic of an optical microscope? Magnification, the numerical aperture of the objective, the size of its objective, the focal length of the objective, or the number of lenses it has. What's the most important of all of these? And Well, hey, yeah, people were listening. Everyone got that one correct. Wow, so it's probably still neck and neck. Let's see who was fastest.
Too simple.
The quickest on the button gets the most points. Oh, IMCF. Ooh, yeah. Let's see where we are. Oh, Stingalicious is still holding on. Okay. It's all to play for. So next question. And that question is, what is chromatic aberration? Oh, yeah, you've explained this one. Let's see if people were listening. The vacuum speed of light is different for different wavelengths. Propagation of the direction of light changes from passing from air into glass. A sample looks fuzzy in a microscope. A lens focuses light of different colors to different. Everyone's there. So I think this is all about speed today. We got a bunch of smart people. So, oh my goodness me. Let's see where we are. Oh, INCF has sneaked into the lead. Okay, next question. And this one is, what is spherical aberration? A spherical lens does not focus parallel light to a single focus. Propagation of light changes when entering from air into glass. A sample looks colored in microscopy image. The vacuum speed of light is different for different wavelengths. Oh, yeah. Yeah, basically focuses to different points. All right. So let's see where we are on the leaderboard now. Ooh, IMCF is out the running. Oh no. Stingalicious is back in the lead. Well done. Okay, and the last question. Now here I had to write lambda instead of wavelength because there's no way of putting symbols into Mentimeter. So, so if you just see the word lambda, it just means wavelength. Okay, so what is the correct expression for Abbe's resolution limit of optical microscopy? D, so the resolution equals lambda over n equals lambda over 2NA, is 2 lambda over n times NA, or D is lambda times n times NA. And yeah, 5 people who know the answer. Well done. Obviously, our very smart people following this week. And let's see who is the final winner. Ooh.
Oh.
Got in there first. I'm not quite sure who's the winner. Stingalicious. Make yourself known to us through the chat, or just give us an email, some sort of contact. We'll send you the Fold Scope, our wonderful prize. All right, well done for everyone for playing that. That was fun. I've never seen such a tight competition.
Next time it's more challenging.
Okay, so back to the questions. So you answered that one about.
There was one very nice question from Bilayer. When we approach angstrom resolution along the z-axis, does the flexibility of the linker with which the dye is linked to the bilayer, does it not then come into play? Yeah, I guess so. But I mean, what we do is we measure really densely labeled bilayers. So what we see, of course, is only the mean distance from the surface. And I guess when the density of labeling is high enough and when the bilayer is homogeneous enough, then this is simply averaged out. You only see the average value. But I mean, you are right. In principle, looking at, let's say, the distribution of the lifetime, we should then at some point see the linker flexibility. We did not look at it. This is it's a good idea. Maybe you should do it.
I think the second part to that was how long do you have to measure for a confocal storm image? Yeah, I mean, for the images, I showed you one example where we look at the chromatin in the nucleus and also at the tubulin network. Yep. And this is, you can do it with a few images per second. So we are talking about something like 1 to 10 hertz. Yeah. It really depends on how bright your dyes are and how much excitation intensity you put in. But I guess, of course, you cannot go as fast as with wide field, for sure. Yeah, you sort of... What is the proportion of distorted PSFs due to blinking bleaching? I don't understand this question, I have to say. What is the proportion of distorted PSFs? Yeah.
What do you mean with this solid PSFs?
Dirk, you can unmute if you want and ask.
I mean, what we observed or still in the beginning when I did my PhD, when we started confocal scanning of single molecules, we saw that molecules were bleaching while we were scanning them, right?
No, I mean,
and therefore the PSF is no longer around, but it's kind of cut off.
Because we are so fast, we don't see that, really. I mean, I can send you raw images. We don't see that. We are so fast. Of course, what we do is now we scan super fast with something like 20 Hertz, and then we bunch image, also we then simply bunch images together. Yeah?
And Joel had.
And then may I just add, because I'm not sure if the first part of the question was really understood. So my question was actually, how long does the recording of a whole storm image basically take for you?
Oh, okay, yeah, long. I mean, something like good question. So I think for the Chroma team, in the order of 30 minutes. Okay. 30 minutes, yeah. But it is now very rough. It really depends. I mean, so I did, I mean, the point is, by the way, I mean, our lateral resolution is certainly not as high as the best wide field storm images, because we don't collect as many photos from one single molecule as you would collect in a wide field microscope, because due to the scanning, the duty cycle is much shorter. Yeah. Yep.
But that would basically mean if you had ways to do the same data acquisition using a camera, that should improve your approach, right?
And we published that. We have one publication where we use a lifetime imaging camera for doing the same thing. It works very well, better than expected. Also, the camera has a quantum detection efficiency of only 5%. It's so, but I mean, we hope that in the future we will see more and more these lifetime imaging cameras like these sputter rays and so on. This will be a game changer, but at the moment,
So maybe fast forward. I've posted just another question into the chat. Would you have time for a follow-up Zoom later on today?
Yes, pleasure.
Cool. Yeah. Let's talk. See you.
I think we're going to have to move on to the next person. We're already over time. Sorry there, Peter. So Joel, do you want to unmute and ask your question if you're still there, or I can? Oh, yeah. Joel.
Yeah, I'm still here. That was a fantastically wonderful educational talk, Jörg. Really enjoyed it.
Thank you.
The physics may go over my head a little bit. In terms of the laser power required from it, imaging, is that more or less for standard confocal or standard Flim imaging?
This is standard. So we use, for excitation, we use only something like diode lasers or white light lasers. So you have a typical excitation intensity in the order, as in the focus of, let's say, 10 to 100 microwatts.
Okay. Yeah, so you're still looking after your cells. You're not worried too much about killing them off while imaging.
No. I mean, this is really, and also we do this fast scanning. This is also some advantage. You are not driving them so much from triplet state into a bleach state. No? So because you scan so fast that they have always the chance to come back to the ground state until the next scan is coming.
Brilliant, thank you.
Okay, well, actually, I have a couple of questions. So I noticed on your graphene version of this that you had a silica and a polyl-lysine layer on top of the graphene.
Yeah.
Is that for adherence, or is that for some other purpose? And does it have an effect, in terms of its dielectric properties?
Also the spacer layer, silicon dioxide on top of the graphene, has basically a negligible effect on this mite metal potential transfer. This is not changed, because this is really the interaction between your graphene excited electrons and your dye molecule. But we use this spacer layer simply to protect or to shield the biological sample from the highly hydrophobic graphene surface. So I mean, I think a cell will not be happy when it sits on a graphene.
Right.
That's all. So it's simply a protecting layer, and it's very thin. So we are talking about something like 5 to 10 nanometer quartz. It's a very thin layer.
Okay, so there are other methods, aren't there, to sort of functionalize graphene in order to get over that hydrophobicity, aren't there? Or is
Yeah, there would be one way, but I mean, we are not chemists, no, we are physicists. So we are
Okay.
buying the graphene from a company, and then we have a transfer process, how to transfer it to the glass cover slide.
Yeah.
And then in the clean room, we simply cover it with quartz. That's it.
Okay, right. And I just have another observation. So anything that's sort of energy transfer, that's a sort of protective effect against photo bleaching, right? I mean, in it.
Yeah, this is indeed, indeed. I mean, when this is what we see. So let's say when we do these single molecule might experiments with different distances of molecules to the gold, what we observe is that the molecules which are closest to the metal, they survive longest. Yeah, because they have a shorter lifetime, so they are more photostable. However, of course, due to the quenching, we also get less photons. So the fun fact is that then the net yield of photons until bleaching is basically independent on the distance. Yeah, the farther molecules, they are brighter, but they bleach earlier. The closer molecules, they are dimmer, but they bleach later. Yeah.
Okay, that's an interesting observation, actually. Okay, so we've run quite a long time over time, equal past, and I think we should wrap up. But I would just like to say thank you very much for such an interesting talk. That was really good.
Pleasure.
People can just wave their Zoom hand or otherwise congratulate you. So thank you very much, and a real clap. Audience of one. So okay. So next week we have another talk. I think that we, Jess, since you're there.
We do, yes. I don't have to hand exactly who that is. I apologise.
Well, anyway, you'll find that coming soon. Anyway, another one, and I would just like to say once again to Jörg, thank you very much for such an illuminating talk, and see you next week.
Yeah.